Hi then, so this is going to be the final video that I do on proteins. Just as a reminder, in the first video we spoke about how peptide bonds are formed and broken. Then we looked at the structure and the different levels of structure in proteins in the second video. Talked a bit about the bonds present there as well. So in the third video, that was the bonds present. And lastly, we're just going to look at how changes to the primary structure can lead to a non-functional protein. Now this is really important and it comes up a lot throughout the first year content and also in the second year content. When we move on to DNA and the genetic code, we'll look at how this is important as well. Okay, so you may remember from GCSE the idea of denaturation. So denaturation is the shape, the three-dimensional shape of the protein being disrupted. And you may remember from GCSE there were a couple of the causes mentioned here. So an increase in temperature, a change to the pH, you may have spoken about in at GCSE. Now, what we need to build on from your GCSE knowledge to the A-level knowledge is the reason why we get this denaturation. And that links back to the bonds which are formed in the secondary and the tertiary structures. Okay, so in the secondary structure, if we remember, we've got those hydrogen bonds. In the tertiary structure, we have the hydrogen bonds, the ionic, and then the disulfide bridges as well. Disulfide bridges or disulfide bonds. So, temperature can have an effect, particularly on these weak hydrogen bonds. So an increase in temperature increases the kinetic energy. That increase in kinetic energy causes those hydrogen bonds to break. Now, if those hydrogen bonds break, it results in a change to the secondary and tertiary structure. Okay, that change to the secondary and tertiary structure is what we mean by denaturation. So the shape of the overall shape of the protein would change as those hydrogen bonds break. Ionic bonds, okay, a change in pH perhaps would have and salt concentration would have a larger effect on the ionic bonds. And then both disulfide bonds and disulfide bridges are particularly affected by heavy metals and the presence of heavy metals and the reduction of the disulfide bridges. Okay, so why is that important? Well, if those proteins are fibrous, i.e. longer proteins that play a structural function in the cell, they may lose their structural strength, they may lose their shape, and that structural property that they have would be lost. If they're globular proteins, such as enzymes, receptors, etc., they lose their shape, they no longer have a very specific and perhaps a complementary structure. It may change the shape of the active site or the receptor site, and therefore they become inactive. So we'll look at that in a bit more detail now. So, a fairly typical and common question that arises in biology is sort of follows this theme. How can a small change in the primary structure lead to a non-functional protein? Now, the question won't always be like this, but the concept or the principle here comes up a lot. Okay, so a change in the primary structure. What do we mean by that? Well, obviously, the primary structure are the sequence of amino acids in the chain joined by those peptide bonds. Okay, it's probably not the best diagram I've ever drawn, but these are, so the primary structure are the sequence of amino acids. A change to that sequence of amino acids in the primary structure means that hydrogen bonds and ionic bonds and disulfide bonds, perhaps, form in different places. Now the examiners really want you to say this. Okay, the idea that the bonds are being formed in different positions. Now we know that the amino acids in the secondary structure, those hydrogen bonds form between the carboxyl groups and the amine groups. If the amino acid sequence changes, perhaps those hydrogen bonds in the secondary structure form in different positions. Now that may change the shape of the secondary structure. Perhaps it would prevent an alpha helix or a beta pleat from forming, or cause it to form in the wrong place. Okay. In the tertiary structure, those hydrogen bonds, ionic and disulfide bonds between the R groups, they could also form in different places, in different positions. So the idea that the bonds, whatever they are, hydrogen, ionic, disulfide, form in different places or positions is really vital. Okay, that will always be one marking point. So change in the sequence of the amino acids is a marking point. Hydrogen bonds or ionic bonds or disulfide bonds forming in different positions is always a second marking point. What effect does that have? Well, it changes the tertiary structure of the protein. Okay, so very commonly this might be a 3-mark question. Okay, with those 3 marks, we've got 1 mark for the change in the primary structure, 1 mark for those bonds forming in different positions, and a 3rd mark for the change in the tertiary structure. Very commonly, this may relate to an enzyme. What do we know about enzymes? Well, enzymes have a very specific active site which would be complementary to a substrate. Okay, so let's go through this. Change in the amino acid sequence in the primary structure means that hydrogen bonds, ionic bonds, disulfide bridges form in different positions. That changes the tertiary structure, which would mean that the active site of an enzyme is no longer complementary to a substrate. Therefore, those enzyme substrate complexes cannot form, and therefore the reactions cannot be catalyzed. That's the way we go if it's an enzyme. Some cells have receptors on their surface. Receptors are often proteins or derivatives of proteins, and therefore they also have very specific tertiary structures. Receptors, however, do not have active sites, but they do have a receptor site. Okay, and that receptor site may be complementary to a particular hormone, for example. So if the question were, what if this protein were a receptor? Well, that would change the shape of the receptor, so it was no longer complementary to a hormone perhaps, and therefore the hormones do not bind and do not bring about the response. Okay, so this type of question comes up an awful lot. When we move on to look at enzymes, we'll return to this, but these are absolutely key concepts in biology. Okay, I'm just going to talk now a little bit about different types of proteins, just to finish off on our finish off this topic. I've sort of mentioned these briefly before. Two main groups of proteins that we need to be familiar with are the fibrous proteins, where the polypeptide chains tend to form in parallel lines. Okay, so we may get parallel polypeptides forming, and then cross-links form between those parallel chains. Now those could be hydrogen bonds, ionic bonds, disulfide bridges between these parallel polypeptide chains. Obviously this is a plural, so this would be a quaternary protein. That means that they can form very long, rope-like fibres with high tensile strength, i.e. they resist snapping. Okay, this idea of high tensile strength, force can be applied to them and they resist breaking. Usually these play a structural role, or they do in fact, and they will usually be insoluble in water. We'll give some examples of these in a moment. Globular proteins, on the other hand, could be tightly folded polypeptide chains. They may be tertiary structures or quaternary. Think about a quaternary one might be hemoglobin. Tertiary structure might be something like an enzyme. They tend to have a three-dimensional spherical shape. And as we mentioned in the bonding video that I did in here, they may have hydrophobic groups on the inside of the protein, avoiding any water in solution surrounding them, with hydrophilic, water-loving R groups on the outside. So hydrophobic R groups on the inside, hydrophilic R groups on the outside, and they tend to be soluble in water. Okay. Now, it's important to remember then that the shape of the protein determines its function. The shape of the protein is derives from that primary structure, the sequence of amino acids in the polypeptide. Some examples, you don't need to remember these, but these are some common examples. So collagen, found in connective tissues and ligaments, tendons and cartilage. Collagen is the most abundant protein in the body. It's about a quarter of all of the protein in the human body. Keratin, those harder structures such as hair, nails, claws and hooves. Silk would be another example of a fibrous protein. So spider silk or silkworm produced by, the silk produced by silkworms, etc. Some examples of globular proteins. Most commonly, we might think of something like hemoglobin with that quaternary structure, so 4 polypeptides in a quaternary structure. This is actually a structure of myoglobin. So myoglobin is just a single polypeptide that contains, or that is found in muscle tissue. And it acts as a store of oxygen in the muscle. So when the muscle is undergoing real strenuous exercise, the myoglobin gives up its oxygen, which delays anaerobic respiration. We'll actually talk a bit more about myoglobin later in the year when we look at transport in animals. Okay. Other globular proteins very commonly might be enzymes or hormones. So enzymes we've mentioned already, very important that their tertiary structure is specific. The active site will be complementary to just one substrate. Hormones, estrogen and insulin. Actually, insulin and let's say glucagon, ignore estrogen there, are both proteins, and they have a very specific tertiary structure which is complementary to a receptor on the surface of the target cells. Last thing that I'll mention here is the test for proteins. Now, we will be doing this in class when we do practicals, and we'll use the Bio-Rett test. So the Bio-Rett test for proteins, take a sample of the protein, mix it with the two Bio-Rett solutions. If no protein is present, the sample will remain blue. That's a negative result. If the protein is present, it will turn purple or lilac, mauve colour, to indicate the presence of a protein. One little common error here is people say that it's necessary to heat the Bio-Rett test. It is not necessary to heat the Bio-Rett test, and in fact you will lose marks. So I'm just going to put in here no heat necessary. Okay, that's everything on proteins. I'll do another set of videos related to this where we look more specifically at enzymes. Thanks a lot.